We are going to help you and guide you through today's program and manage the Q&A with our speakers. So, today's symposium is part of an ongoing collaboration between the DOC and EWOC to highlight outstanding research in organic chemistry, while also promoting a more inclusive, supportive, and empowering environment for women and allies at all career stages. So, you can see here a slide showing some of our the founders of the EWOC conference a few years ago. So it started off in a few years ago. It had to go virtual for a little while, then it went in person. But these women, they have been fantastic at moving along women in organic chemistry and creating this space for us. Next slide, please. So you can see we've had a number of meetings, even during the pandemic, that we had to go virtual. So uh, they are registrants that are online and in person. The if you can see in there that the conference takes place in both coasts. It alternates. So in 2025, it was in Chicago, and it was extremely successful. So we have representation from EWOC's chapters and different panelists. Next slide, please. So this year it's going to be in San Francisco. So we invite you to attend either, oops, let's take it out of the way, either in person or virtually. You can register for the conference at ewochem.org. That should be available soon, and it's going to be at Genentech, the original biotech company. So we're grateful to have you here and uh, to feature, the talks that not only showcase cutting edge science, but also reflect the leadership and impact of women in our field. So, Layla, back to you.
Thank you, Monica. So if you have any questions, well, we're excited to feature the talks that showcase not only cutting edge science, but also reflect leadership and impact of women across the field. First, I'd like to introduce a few DOC events going on this year. Of course, we have our March meeting, ACS. We have award symposia featured here on this slide. Make sure you don't miss them. We also have organic joint symposia, Academia 101, new this year, Biopharma 101, a bunch of other symposia in collaboration with MEDI, YCC, Comp, and Phys. Please don't miss them. Deadlines for nominations of technical achievements in organic chemistry and the Industrial Early and Mid-Career Investigator Awards are February 3rd. And please contact the co-chairs, Elizabeth Swift and Vanessa Marks, for the nominations, and Steve Wyszynski and Steve Silverman as well. The Graduate Research Symposium is going on. This is a picture of it at Emory University this past year. And the application nomination process for this year is March 10th, 2026. So please don't miss it. Lastly, we have our Summer Undergraduate Research Fellowship Awards, and the deadline for applications is February 17th. This is an awesome opportunity for undergraduates to receive $6,000 in support for their summer research, an industry mentor, a site visit, a Meet the Sponsors Symposium and Career Panel, and a virtual research symposium are all parts of this prestigious award for undergraduates. So please make sure you have your undergraduates apply. We have a virtual symposium going on next Wednesday, so please don't miss it. Christian Malapit and Cade Harper next week. And I'd like to take this opportunity to thank our inaugural DOC Virtual Symposium speaker, Professor Sue Sim from last year, who gave an awesome talk, and to introduce this today's first speaker, Christina Woo from Harvard University. I'll remind you that we will have a 5-minute window for questions after each talk, and please use the Q&A feature. Christina is professor of chemistry and chemical biology at Harvard University and an affiliate member of the Broad Institute. She obtained her BA in chemistry from Wesley College in 2008 and conducted undergraduate research in the laboratory of Professor Dora Carrico Moniz. She obtained her PhD in 2013 from Yale University under the guidance of Professor Seth Herzon as an NSF postdoctoral fellow in the synthetic and chemical biology studies of diazofluorine anti-tumor antibiotics. In 2013, Christina joined the laboratories of Carolyn Bertozzi at the University of California, Berkeley as a James Coughlin's Child postdoctoral fellow and continued at Stanford in 2015 as a Burroughs Wellcome postdoctoral fellow, where she developed a mass-independent chemical glycoproteomics platform for the identification of non-templated post-translational modifications. Christina joined the faculty at Harvard in 2016, and her independent research focuses on how small molecules influence protein function and biological signaling using chemical biology approaches. Christina, with that.
Yeah, thank you, Leila, for that very kind introduction, and to both EWOC and the Division of Organic Chemistry for putting together this webinar series, and for inviting me together with Laura, one of my own personal heroes, or someone I closely follow and look up to in the field. And at least for putting me first so that I can get it, enjoy her talk more as a result. So let me just share my slides here. And also to follow on what Leila said, I put together a talk that's a little bit new. I apologize, it's not going to be as organic chemistry focused, but that was in part motivated because I see some of the people here that I recently saw at in the Division of Organic Chemistry over the summer at the fall ACS meeting. So I thought I wouldn't want to bore them. So I put together a talk that highlights some of our ideas in rewriting protein modifications. I used this talk title for many years from the perspective of both the chemical induced proximity thinking that we can induce this type of modification as desired, but also really as a double entendre in a way that I like to think that we are expanding the types of protein modifications that we think about in our biology. But as a result of, I hope that the talk actually won't take the full time. And so I would actually like to invite the audience on the webinar to think about questions. I think one of the strengths of the EWOC community and the organic chemistry community is being able to bring together and highlight women in organic chemistry, and would be happy for you to think about any questions that is about both the science, but also anything else, and maybe anything else. I don't know if that gets taken out of the actual recording, but yeah, I remember actually being part of the EWOC in-person seminar back in 2021. So I'll try to benchmark a little bit the work and progress we've made since then for the community. So as I just described, my group really uses the tools of chemical biology, including chemical probes, protein engineering, and proteomics, to inspire how we think about regulation of cells through protein interactions, and specifically the chemistry on proteins in the form of protein modifications, and then how we can co-opt that with small molecules. We've thought about a number of different modifications to date, but
one of the main arcs of work in our lab has really been now focused in on a protein called cereblon. I'll introduce that one on the next slide. Really inspired by a lot of induced proximity targeted protein degradation findings to date with the thalidomide system, leading us to understand new biology in terms of how cereblon's endogenous substrates are being regulated. And that'll be the main arc of the story I wanted to share with you today as an update there. But in tandem with that, a number of the efforts in our lab have also thought about how we can use this new biological understanding to drive new probes and therapeutics, and I'll just try and highlight that a little bit at the end as well. To unite my talk, and I think really at the core of what I will be sharing with you today is chemistry of the amide. And I think from an organic chemistry perspective, this is, the number one functional group that's synthesized. In fact, it's the last reaction that I did in the lab myself. And so it's, that's just how easy it is to be able to be synthesized. Someone like me can still make molecules like this. And I think when, from an organic chemistry perspective, why it's such a popular functional group to synthesize was because it is so thought to be so stable. And that's one of the ways that maybe we think about why the amide is used as a backbone for proteins as well. But what I hope to share with you today is actually a lot of the chemistry that amides can promote, and both in terms of their nucleophile, electrophile chemistry, the fact that they absorb equivalents of ammonia, release them, and help shuttle that around our bodies. And so I'll try and draw out the chemical underpinning our chemistry first, thinking in the story that I'll tell you today, but it's really united by thinking about all the ways that amides can function in our bodies. So the star of our lab is this protein here called cereblon. This protein was first discovered in 2004 from a genetic study associating cereblon with a normal neurological development. And what the authors of this work, Higgins and coworkers, had found or identified at the time was point mutations in what's now known to be the thalidomide binding domain of cereblon, followed on by a subsequent study from another lab more recently in 2017 with another human genetic mutation that is associated with normal neurological development. And when it is mutated, that would lead to deleterious effects, including seizures, intellectual development delays, and impairment, and self-mutilation effects in the more as well. At the time of the first description of cereblon, it wasn't yet known that this was part of the thalidomide binding domain, but we now know since then and the advances since then is that cereblon is a fairly conserved protein, fairly far back all the way to plants. But the thalidomide binding domain, the part that will be the star of the show today, is conserved even further back. And structural basis of these interactions of this protein with ligands like thalidomide now show that the protein and this domain has these three domains. And much attention has been put to understanding cereblon, initially from its role in intellectual disability, but now more recently because of really seminal discoveries that molecules like thalidomide, which have their own storied history in medicinal chemistry and pharmacology and the clinic, and are now used, and their derivatives are now used for anti-cancer medications, amongst many other applications, is that it was the finding that this compound interfaces with cereblon, and that's how a lot of the mechanistic basis for these compounds' activities emerged. And following on that was the finding that you could take thalidomide and so readily derivatize it into additional molecular glues and PROTACs for targeted protein degradation, and really driving a new wave in the induced proximity field more broadly. So a lot of these findings, or initial ones, now back in 2010, 2014, about a decade and change ago, have now revealed this overall picture that this molecule thalidomide and lenalidomide interface with cereblon. And when they interface with cereblon, they have this exquisite ability of inducing proximity with a number of different substrates in different contexts. And in some cases, this can induced proximity event promotes ubiquitination protein degradation, often of proteins that are otherwise very hard to ligand. This work inspired efforts in our group, along with structural efforts in other labs, to reveal that cereblon is endogenous substrates are recognized through a chemical motif that is really very much inspired and mimicked by the thalidomide derivatives, thalidomide derivatives through this glutaramide ring. And so now a few years ago, and this was where we were last time I really spoke to the EWOC community in specific, was really starting to understand that thalidomide and lenalidomide, the chemical underpinning of the warhead that is used to bind to cereblon, is this glutaramide. And this is an amino acid derivative, a dehydrated form of either asparagine or glutamine. And we came up with this model where endogenous substrates are recognized by this C-terminal cyclic imide, either from asparagine or glutamine amino acids, and this would promote ubiquitination protein degradation. At this time, and this really inspired us to start to look more carefully at where these modifications were coming from, and the organic chemistry, frankly, of underlying the formation, and what kinds of substrates were carrying them. In parallel with our chemistry first approach, focusing in on the molecular basis of the interaction, a number of other studies in the field had started to, from biological models, it started to reveal that there were a number of native substrates of cereblon that were really revealed in a number of different studies. It's impossible to put all of the citations on this single slide, but hopefully we'll be able to review that soon. And what was really intriguing to us when we kind of took a look at these substrates was many of them contained a C-terminal asparagine or glutamine residue. And this started to make us wonder whether or not that was a chemical event that was giving rise to this post-translational modification, and if we could make that connection. The second star of our show is this protein here, glutamine synthetase. And this is one of the substrates that had been annotated and associated with cereblon pretty early on as one of these endogenous substrates. Glutamine synthetase its primary biological role is to take glutamate and convert it to glutamine by absorbing an equivalent of ammonia, using a little bit of energy through that ATP. And most of the time we think about glutamine synthetase as really helping take glucose through the TCA cycle, parts of this that gives rise to glutamate, and convert it to glutamine as a feedstock for protein biosynthesis, ammonia detoxification, pH balance, depending on the part of the body that it's involved in. this is one part of the metabolic pathway that can go in this direction. But in our bodies, we also eat a lot of food that's not just glucose. We also eat a lot of glutamine. And so in normal cellular metabolism, but also especially in dysregulated cellular metabolism, especially in glutamine-dependent cancers, extracellular glutamine is actually the primary source of this amino acid, which gets uptaked and pushed in the other direction to glutamate. And in this way, support ATP biosynthesis as well as protein biosynthesis, glutathione biosynthesis, and redox homeostasis. And so this protein and the kind of regulation in the forward and especially in the reverse direction is actually product inhibited. So glutamine can inhibit glutamine synthetase, both in terms of its immediate enzymatic activity, but also promote its degradation. And the E3 ligase, or the proteins that take this out, is one of is an open question, and there are probably multiple E3 ligases that participate in glutamine synthetase degradation. The protein itself is part of a pentamer that forms the active complex. The catalytic active site is actually at the interface of the homopentamer. One of the ways that glutamine synthetase is inhibited is by actually the forming a filamentation event with glutamine at the interfaces at an allosteric site. I think this is a really cool story that just came online from the Fraser lab, and Green. But this doesn't necessarily directly lead to degradation. There's other pathways involved there. And cereblon is the primary E3 ligase that's been associated with glutamine synthetase previously from the Deshay lab at the N-terminus. As you can see, it's somewhat buried here in the multimer, indicating that there's likely the kind of formation of the N-terminal degron must happen through some kind of monomeric intermediate, I would think. But the story that I will tell you about here is really adding to glutamine synthetase regulation through now the C-terminal tail, and that's highlighted here. And specifically understanding now the C-terminal degron chemistry that is evolved. So coming back to cereblon and our understanding of the molecular basis of cereblon, recognition of substrates. This was a really important question to us, both from really characterizing where that post-translational modification is coming from, but also starting to really querying it from the medicinal chemistry or the pharmacological rationale of thinking of why, whether or not these substrates are being impacted during therapy or when people try and co-opt cereblon for therapeutic desirable events, and is the endogenous substrate system actually playing a positive or negative role in the therapeutic outcomes. So we hypothesized that the post-translational modification, the C-terminal cyclic imide, could come from non-enzymatic sources or enzymatic sources, and that depending on where those sources were coming from, it would lead to the different substrates and pathways and potentially different contributions to therapeutic outcomes. We really initially focused in on the protein damage mechanism, and this is something that was very much inspired by another form of protein damage, the spontaneous deamidation chemistry, which has both similar chemical and biological hallmarks. The chemical hallmarks are that the chemistry of formation of the cyclic imide non-enzymatically involves a side chain amide attacking a backbone amide, and that would reveal the cereblon degron. By contrast, in parallel, as deamidation involves the backbone amide attacking the side chain amide, release of ammonia, and that would have this internal cyclic imide intermediate that would just proportionate to aspartate, but primarily isoaspartate. And the primary kind of difference in the in kind of what happens here is that the protein backbone has been really permutated to have a major change in the isoaspartate product. And this isoaspartate product is thought to be fairly deleterious, requiring this protein carboxymethyltransferase to methylate it, promote a reformation of the internal cyclic imide so that it could disproportion to aspartate over time. So that was the chemistry that really inspired us to think about the non-enzymatic forms of cereblon digron formation. Given that it was happening, in a paralogous type of chemistry in much more frequent observation through deamidation, and then the biology was that PCMT1, this enzyme that methylates the side chain isoaspartate, is also really important in the brain, and so it reminded us of cereblon, especially due to the phenotypes of epileptic seizures. And in the case of PCMT1, it is a bit more severe in onset and drives premature death in animal models. But this didn't inspire us alone, and there were some open questions that we, on reading the papers, and that were clearly noted by the investigators of this field, that this, was a somewhat unfulfilling model in the sense that the disproportionation between aspartate and isoaspartate is about 70% of the time on average you get isoaspartate. So this is a pretty poor correction mechanism. It has to go through this process a number of times to really get to primarily the aspartate, although they could show that they were able to recover enzymatic activity of the protein, whatever the model system was in part. But then the maybe bigger kind of unfulfilling part of it was that this is still a partial repair because you're not going all the way back to asparagine. You're still at aspartate. And so this intrigued us, and we started to work with this enzyme, just trying to understand this, parallel isoaspartate chemistry, how it may or may not reflect the chemistry that we were observing with cereblon and its protein damage recognition mechanisms, which we published earlier last year, I guess, about a year ago. But that through that work, that also led us to start thinking about how is this system potentially actually directly involved in enzymatic formation of cereblon substrates. And for this, we assembled an amazing team of collaborators, both within my lab with Zhenguang Zhao, Wensheng Su, Ethan Feng, and Hannah Lloyd, together with collaborators in structural biology from great contributions from Xi and Cao and Ning Zhang, on the structural end, and Alba, Pablo, and Manuel Guzman's group with in vivo models of cereblon knockout mice to really help us connect the functional annotation of the enzyme formation to cereblon substrates in animal models. And I apologize, just didn't get a chance to get their pictures here on this slide here. So with this team, we were able to make the following connections, which started from a really basic question. Do cyclic imides form on the C-terminus of a protein, either at asparagine or glutamine residues? And that was, again, coming back to the fact that in independent biological studies, proteins had been associated with cereblon, but the molecular basis wasn't really clear. And in the majority of these studies, although some of them had started to hone in on the C-terminus, but a specific connection between a C-terminus asparagine forming a C-terminal cyclic imide hadn't really been understood yet in eukaryotes, in us. And that's because, making these molecules, having a peptide and just having it sit around, you're going to wait a really long time. And so the non-enzymatic cyclization is just not observed under physiological conditions. So that made us think there has to be an enzyme that can do this. And after thinking about it, you can find one example of enzyme like this in bacteria, the MCCB pathway that was annotated by Chris Walsh. But it did it did this chemistry through a phosphate, but then it did a little bit more and a little bit too much by reopening it. But that enzyme system just doesn't exist in our biology. So we were looking for a different enzyme. And that drew us back to PCMT1, eventually was thinking that this isoaspartate modification kind of reminded us eventually of a C-terminal residue of a protein. Where the side chain carboxylate about the same pKa, we hypothesized, and the primary difference here was that the backbone of the protein in this case, or quote unquote side chain amide event would just have a n equals r for the rest of the protein, or n equals h in the context of a C-terminal asparagine or glutamine. So we thought maybe PCMT1 could also recognize C-terminal asparagine and glutamine residues and methylate those. So we purchased, and it just hadn't been tested. So we purchased a series of peptides that represented each of these proteins that had been previously associated with cereblon. We subjected them to PCMT1 when observed and monitored by mass spectrometry, whether or not they would form the C-terminal cyclic imide. And so what we found was that we quickly focused in on glutamine synthetase. This was the one that converted to the greatest degree. We also could see conversion of APP, but methylation of the C-terminal glutamine residues, but not necessarily actually cyclization. And so this led us to focus in on glutamine synthetase, again, this protein that's making glutamate to glutamine in our bodies. And this is the mass spectrometry chase of the C-terminal glutamine pentamer going through conversion in the presence of PCMT1. It's consumed. You see the methyl ester form, so that first intermediate promoted by PCMT1, and then over time that forms the cyclic imide. And the enzyme eventually stalls out in vitro. So by measuring the initial kinetics through KCAT and KM for the C-terminal cyclic imide, as compared to its previously and primarily annotated substrate to date or isospartate, what we see is that the enzyme is about 20-fold so the KCAT and KM that we measure is about for isospartate for this peptide here that had been previously annotated by Steve Clark as a really a prime substrate for PCMT1 peptide sequence. It's a KCAT or KM of about 2.2 micromolar, and the C-terminal cyclic imide is essentially about 20-fold worse in terms of, or slower in terms of PCMT1's taking it up as a substrate. So this it's not the preferred substrate of PCMT1, but it also didn't totally deter us because PCMT1 has such a broad range of isospartate substrates, and some of them have KCATs and KMs in this region. So we continued to take a closer look. We also showed that this is, depending on the active enzyme, the inactive enzyme doesn't form the cyclic imide, and you, of course, need the asparagine to cyclize. Together with the support of Ning Zheng's group from Xi'an, we were able to get a crystal structure of this degron in the cereblon binding pocket, showing really the contacts highlighted here in terms of the amino acids that are involved in contacting this C-terminals of glutamine synthetase, and all of these amino acids are necessary and conserved in evolution. So from there, we took a deeper look at the full process and on glutamine synthetase, and were able to gather evidence at every stage. And I'll just show you a few highlights here of the from mass spec in terms of really mapping that the cyclic imide is forming in a PCMT1 dependent manner for this initial phase. Okay. Then showing that it will bind to cereblon when it's there, when the cyclic imide is there, and then this will promote in vitro ubiquitination, but also protein degradation when you introduce GLUL into a cell via electroporation. You can see its decrease only in the presence of PCMT1 when it's treated with PCMT1, indicative of the requirement of the cyclic imide. And excitingly, endogenous glutamine synthetase levels when you knock out either cereblon or PCMT1 also elevate the elevate to a similar degree the amount of glutamine synthetase. But what you can see is that we're getting a shift here from 1 to about 1.3, 1.4 of the protein levels in this is in HEC293 T cells. And so to help us better visualize this protein modification, which is a substoichiometric protein modification event, we had a parallel program really driven by Hannah Lloyd in our group. With its own set of amazing collaborators, to engineer a cereblon derivative into what we called a Cerebody that would enable us to study cereblon and detect its substrates more readily through either protein enrichment or Western blot use of this, what we thought of what could be an antibody. And so not walking through all of the details of the engineering, frankly, we got lucky with other protein engineering work that was happening in the field, especially from Alessio Ciulli's lab, showing and being able to use constructs like his Cereblon MIDI, enabling us to create a more stabilized version for our approach with this Cerebody. And now having a really facile expression system with tight binding kinetics and enabling the visualization of C-terminal cyclic imids by Western blot, whereas previously we were confined to mass spectrometry. This made it a lot easier for us to now enrich glutamine cyclic imide, so GLULCN, the PTM, the post-translationally modified version of this protein. And so after the Cerebody enrichment, you can see really clearly that this cyclic imide form is present when you treat cells with lenalidomide. That should block cereblon, promote the or prevent the degradation of glutamine synthetase in this modified form. The enzyme knockout should prevent formation of the cyclic imide as well, and that's what you see. And cereblon knockout prevents the degradation of the cyclic imide form, and you see its rescue here as well. And that's not just in HEC293 T cells, but also in a number of different cell types, both in neurons, but also in mice, in mouse cell lines. And so this led us to turn to Manuel Guzman's group, where we were really excited to be able to tap into mouse hippocampi amongst other parts of the animal to now look at, by global proteomics in this instance, cereblon knockout mouse. You can see cereblon is reduced. But now that we know what we're looking for, we can really see that enzymes like GLUL are upregulated. Other enzymes as well, like PPA1, are also upregulated. And using Cerebody enrichment methodology, we can really clearly visualize the cyclic imide forms of those as being upregulated. We now looking at these 4 proteins, cereblon, CMT1, GLUL, glutamine synthetase, and pyrophosphatase, an organic protein, pyrophosphatase, that's PPA1. And specifically looking in the brain, that's where a number of the kind of convergent biological annotations of these proteins had been annotated in humans. And taking a look in this mouse data set for where these proteins are co-expressed, you can see that they have a cluster here in this region of the mouse brain on this map. And taking a look at which cell types those are, they are glutaminergic neurons. And these are the neurons that are involved in the excitatory response, coming back to that seizure phenotype that had been previously associated with cereblon and PCMT1. And so we investigated whether or not cereblon knockout mice have a predilection or challenged those mice with seizure induction. And so we took kainic acid. This is a molecule that induces seizures, and monitored the resolution of the seizure. And what you can see is that in the wild type mice, it goes from an induction of the seizure. There's a phase 2 that in phase 1 and phase 2, you have a kind of seizure starts to resolve and a decrease in phase 3. Whereas the cereblon knockout mice have an initial phase 1 very similar, but they have a much harder time of resolving the seizure. And perhaps this helps explain in part the human genetic associations that are in the literature now through dysregulated substrates and associates with dysregulated substrates like glutamine synthetase. Looking specifically at the metabolite levels for glutamine synthetase, you can see that in the wild type animal, there's a decreased amount in the glutamine levels after chinic acid induction, and this is tempered on cereblon knockout, and the amount of ammonia is increased, and this has not been, this is decreased between the wild type to cereblon knockout mice, but the kind of response upon seizure induction is not altered. So there's still quite a few more questions that are involved here, and I'm not 100% sure how I'm doing on time. So I hope that I will have left time for questions. Overall, the story that I really shared with you today was really associating PCMT1 to glutamine synthetase and cereblon C-terminal cyclic imide formation and proteasomal degradation of enzymes like glutamine synthetase. We primarily looked at this in the context of the brain, but there are a number of other tissue roles for these proteins, and that's a space that we hope to understand further in the next phase of our work. We also hope to better understand how thalidomide, lenalidomide are interfacing with this pathway. Is it also part of the glutamine synthetase feedback mechanism, as well as better understand the original intent of some of our studies with PCMT1 is the isoaspartate role of PCMT1, is it playing a role back to glutamine synthetase? And perhaps one of the uniting features of this is this equivalent of ammonia, and whether or not there's a mechanism that connects this here. The work I share to do today is on glutamine synthetase and a few, PPA1. What's really interesting to us when we take a look at the C-terminal N, glutamine synthetase itself is conserved, it's one of the most ancient enzymes, but C-terminal asparagine really emerged from fish, sea to land animals. And so we think maybe this plays a role in the way that we process nitrogen for elimination. So we're starting to take a look at PCMT1 and how far back it recognizes C-terminal asparagine is compared to isoaspartate. But you can see based on just the check marks alone, cereblon, PCMT1, and thalidomide binding domain are conserved even further back. And so maybe there are additional substrates that intersect with this pathway that we're looking at in the future. So these are some of the future directions that are exciting us right now on this vein of work. There, we just wanted to come back at the end here to highlight how the biology that I just shared with you gives rise to new chemical opportunities. And so, for example, the chemistry that I showed you with PCMT1 today, in terms of the methyl ester and cyclization, is also a chemistry that Craig Cruz recently showed in his work in a paper in JACS that demonstrated that you could come in with a methyl ester C-terminal asparagine and then have that cyclized to form targeted protein degradation ligands. And I hope to see those kinds of applications in the kind of more small molecule driven area, going with PCMT1 as an enzyme as one potential use of the science that I just shared with you. But I thought I would just leave here to highlight our work today on cyclic imide warheads from a more targeted protein degradation perspective. This was our initial inspiration, was how we could to discover and study cereblon biology was to use PROTACs, these bifunctional molecules for targeted protein degradation, substitute the cereblon warhead, and really characterize and study how those ligands can co-opt cereblon as well. This is an example molecule of one of the compounds that we've synthesized. In this work, we show that cereblon will recognize a number of different cyclic imide warheads, which are basically the C-terminal cyclic imide combined with an amino acid at what we would call the N-1 position. And so we did all of the natural 20 amino acids here. This also, and we could see that the degradation outcome would be tuned based on the side chain here. But what this also inspired in us was thinking that the side chain amino acid could also be used as a new prodrug strategy. We thought to glycosylate in its specific because we still we draw a lot of inspiration, which I forgot to highlight, from the glycan field. In terms of the thinking about cereblon and its metabolic role in biology, but also in this case as a potential use for metabolic gate to targeted protein degradation, in this case by glycosylating the side chain of the PROTAC and promoting blocking cereblon degradation until deglycosylation inside the cell. Okay, with that, I hope I've impressed you with amide chemistry in all its different forms. And there's more that we're excited about to come. And finally, and most importantly, the number of collaborators, both in the lab and outside of it, that have contributed to this work, funding from a number of sources that enabled the science that I shared with you today. And with that, I will close and thank you again for the opportunity to share our work here, and we'll be happy to take any questions. I hope I left enough time. Yeah, thanks.
Thank you, Christina. That was absolutely wonderful work. I don't see any questions right now in the Q&A. If you do have questions for Christina, please drop them in the Q&A. In the interest of time, it is my great pleasure to now introduce our next speaker. Professor Kiessling received her undergraduate at MIT, where she performed undergraduate research in organic synthesis with Professor Bill Rausch. She received her PhD degree in chemistry at Yale for her research with Stuart Schreiber, and she was an American Cancer Society postdoctoral fellow with Peter Dervin at Caltech. She joined the faculty at the University of Wisconsin-Madison, where she became Steenbrock Professor of Chemistry and Lawrence Anderson Professor of Biochemistry and Director of the Keck Center for Chemical Genomics. In 2017, she returned to MIT as the Novartis Professor of Chemistry. She's a member of the American Academy of Sciences, American Academy of Microbiology, the American Philosophical Society, and the National Academy of Sciences. She's founding editor-in-chief of the journal ACS Chemical Biology, and it is my pleasure to welcome Professor Kiessling to this symposium.
Thank you so much. And thanks, Christina, for your beautiful talk. It's great to be here. I also want to thank the division for its support and of younger faculty and students. It's been really great to see how that's really grown over the years. So I'm going to talk a little bit about the role of carbohydrates in health and disease. And this is something that I know is also near and dear to Christina's heart. This shows two cells coming together, and we tend to think about the proteins on the surface of the cell as having these critical roles, but you can see this is the carbohydrate portion really coming together. And this is a picture on here of a mouse small intestine where the bacteria are stained by their carbohydrates, and you can see them all here in red. So I'm going to talk a little bit about protein-carbohydrate interactions in those contexts. And I thought I'd begin where I began to think about these. I was really intrigued by natural products. And if you look at natural products, many of them are glycosylated, and those glycans play critical roles. So I was captivated as a graduate student by calicheamycin. It was the target that I was working on for synthesis, and you can see I was working, of course, on the cleaving module, but the recognition module that actually is involved in DNA recognition. You can see here, this is a structure from the Kahn lab. And you can see that this sugar is really critical for the recognition of the DNA. And in fact, there have been now a number of antibody drug conjugates. Some of the first were actually calicheamycin conjugates to antibodies that happen to target CD22 or CD33. And these are proteins upregulated on B cells, and these are actually targeted to these proteins that in fact themselves bind carbohydrates. So the antibody actually targets a carbohydrate binding protein, which just shows how advantageous these carbohydrate targeting agents can be for therapeutic purposes. And in fact, that is because the sugars that we put on our cells really report on the cell identity and state. So here I've shown some sugars to highlight, right? I think it's very well known that blood groups differ by the sugars you put on your blood cells, and that there are tumor antigens. And this one is one that both the Wong group and the Danishevsky group, as well as other groups, were interested in using as a cancer targeting agent. And of course, I always like to put this up, that sperm egg binding is mediated by protein carbohydrate interactions. What I've shown down here, though, is that it's not just the identity of the cells, but it's the state of the cells. So these A, B, and C are 3 different cancers where the presence of different carbohydrates within a tumor varies. And these carbohydrates here are depicted by these sorts of shape recognition elements, but you can see this is a cervical cancer, there's a breast cancer, and a gastrointestinal cancer, and you can see the difference in the glycans. And so the red shows differences from normal tissues. And so we know that these are really critical, but we don't know very much about how they're recognized and how physiologically we can take advantage of them. And that's really what my lab is very interested in. So I use this to show every cell we've ever looked at on the planet has a carbohydrate coat, including enveloped viruses. And if we could better understand recognition, we could, for example, deliver drugs or co-opt immune recognition. And both of these are used in that antibody drug conjugate. We can, perturb recognition, or and if we better understand this, we can really tackle human health and disease from a different viewpoint. So that's what we're interested in. And so a place to start is to think about what natural proteins that we have in us recognize. And that's been the take that we've used. So we know in us, we have about 200 human carbohydrate binding proteins. And these proteins are critical for our immune function, and for really distinguishing between normal host cells and abnormal cells. So that abnormality could be cancer or it could be a pathogen. And so we have been very curious about what these do. And these proteins kind of come in 2 flavors. We have soluble lectins. So lectins are, again, carbohydrate binding proteins that are not antibodies. And we have them found at barriers where we encounter foreign entities, for example, our gut or our lungs. But we also have them even in places where we don't fully understand their roles, like the brain. And so these are intriguing proteins, and we've been really curious in de-orphaning them and figuring out what their real targets are. So that's what I'm going to talk about today. And as I said, in many cases, we don't know their roles. In fact, a lot of them, their ability to bind carbohydrates hasn't been explicitly shown. It's just inferred based on their structures. And I mentioned a lot the soluble lectins, but we also have transmembrane lectins. And that's where the CD22 and CD23 that I already referred to fit in. These are found mostly on our immune cells. And their job is really to detect normal and abnormal glycosylation, again, so that we can then use that information to train our immune system to recognize and eliminate foreign pathogens. So I'm going to just tell a story about one of these proteins, well, really two, that we've been working on because we have a lot of, it's been really inspiring our ability to think about recognition. So I'm going to get back to some more physical organic chemistry in a second, but just to set up why we're interested in them. These are proteins that are found at these mucosal barriers. We're especially interested in human intellectins, and they're soluble proteins. And again, they're associated with inflammation. So genomic data links them to Crohn's disease, ulcerative colitis, asthma. So basically, diseases of the gut and the lung. And so we didn't know when we started very much about them, but as I said, they're found at these microbial barriers. And they're found really throughout chordates. So chordates, organisms with a spinal cord, which even includes the sea squirts, all the way to us. And if you don't recognize this, this is Rosalind Franklin. I thought she's a good EWOC representative of Homo sapiens. So what I showed you initially was work done by Amanda Dugan, a postdoc in the lab. And what she showed is we could use this protein to specifically light up bacteria in the gut. So this is a small intestine of a mouse. And you can see these are the intestinal villi. This is the kind of mucus layer here. And here you can see the protein is binding not to any of our cells, but only to the microbial cells. And that interaction is calcium dependent. So if you use EDTA, a chelator, you don't see binding. And so how could some carbohydrate binding protein recognize only the bacteria? And so we first looked at this using arrays, and there are great tools to study protein carbohydrate interactions. You can use a mammalian array, which has defined sugars, many of these chemically synthesized, or a microbial array. Many of those have been isolated. And so what we determined is this protein really didn't recognize any of the mammalian sugars. It recognized only microbial sugars. And all of the sugars that it mostly bound to are shown here. And so we were able to understand this by solving the structure of the protein. So this is the first structure that was determined of this fold. It's a trimer, as many carbohydrate binding proteins are. It had 3 galactofuranose residues bound. Those were the sugars we added. It has calcium in the binding site, and it binds to the galactofuranose via this 1,2 diol. And that is actually a conserved feature of all of the different ligands it interacts with. And it binds that 1,2 diol because it has a sort of aromatic box that prevents it from having the binding to a diol on the ring. You can see the diol coordinates to calcium, and over here is one aromatic, and over here is the other. And so it's just kind of boxing that in. And what we could see when we looked at this is that all of these agents bound the same. In fact, even things that had a carboxylate, a much better calcium ligand, would bind through this little diol. So I want to just take a step back, and you might not be so interested in carbohydrate recognition as I am, but carbohydrate recognition is fundamentally different than the kinds of interactions we tend to look at. And so the carbohydrates are basically highly polar, like organized water. And so I've been long interested in how proteins actually recognize these. And I already mentioned to you about calcium binding, et cetera. And we had actually started in my lab by looking at carbohydrates that are also calcium binders that recognize polysaccharides. And we knew that this was the glycan ligand for L-selectin. Initially, this was shown to be a weak ligand. And if we sulfated this position, which was critical for recognition based on biological data, we saw only a threefold enhancement of activity. And so this actually got my lab really interested in mimicking the features of these mucin-like proteins that the carbohydrate lectin bound to. And so at the time, we were really inspired to make mimics that didn't have natural backbones, that had polymeric backbones, by the work of Bob Grubbs. And this is Bob with my daughter many years ago. But I love this picture because it reminds me of what an amazing scientist he was. I miss him dearly. And I'll just say that losing Chuck Casey just recently, another amazing organometallic chemist, has been rough on those of us who have looked up to them so much. So Bob's catalysts and the Schrock catalysts have been remarkable in their chemoselectivity, and you can use electron-rich or electron-poor alkenes, depending on the catalyst, to control the end. So you can put unique end labels, something that we did. And then a great thing about many of these catalysts is that you can control polymer length by just controlling the ratio of monomer to catalyst. And so in this way, we could make different lengths. And we did these experiments a long time ago, but we made these mucin mimics that either had a hydroxyl group at this site or a sulfate group at this site. And again, this was really a depressing day when we saw only a threefold potency. But when we make these into polymers, we saw a 10,000-fold difference in potency. It was really remarkable. And in some ways, you might ask, what accounts for this dramatic increase in potency? Is it only multivalency? It's actually more than that. And what we had thought is there was some data in the literature that suggested if you clustered these proteins, you would activate a protease on the surface of the cell that would cleave them from the cell. So you see, though it wasn't clear, my talk is actually related to Christina's in an odd way, but we were able to actually activate this protease by clustering the proteins on the cell. And I think now the Wells group has actually done some beautiful work coapting cell surface proteases through bifunctional or multifunctional agents. But I think this is one of the first examples where we could take advantage of this protease to actually downregulate an inflammatory immune response. And I think making specific ligands is going to just generalize this strategy. So I want to just continue a little bit with how one might then recognize polar ligands in the first place, right? Obviously, if you have a series of weak interactions, you can take advantage of multivalency. But what if you do not? And so one of the things we wanted to look at is how you might bind carbohydrates that have, again, just organized water. And we thought bifurcated hydrogen bonds is one such strategy. And there you can imagine gaining through entropic release of water, right? You have one molecule as opposed to two molecules interacting there. Calcium coordination, another way where you have a chelator and you release 2 molecules of water for binding. And then something else that was in the literature was aromatic interactions. And it was thought that the side of carbohydrates are nonpolar, and so you would get these sort of nonpolar interactions through the aromatic interacting with nonpolar CH bonds. And so we did a bioinformatic analysis that showed actually it's not these bifurcated hydrogen bonds necessarily, or straight up nonpolar interactions that drive binding, but really these aromatics, especially tryptophan. And the preferences we saw are very reminiscent of the beautiful cation pi interactions that Dennis Doherty studied. But of course, we don't have a cation. We have only CH bonds. And so we were really curious about the nature of this interaction and whether there were stereochemical preferences of the carbohydrate. And so what we saw when we began to look at this is that there were stereochemical preferences. So for glucose, there was no real preference for binding aromatic residues to one face or the other. But to beta galactose, all of the aromatic residues were on a single face. And this is best illustrated by this picture. You can see these are all the tryptophans in the database, and they all line up on one side. And if you look at the side that they line up on the side that we organic chemists would predict as having CH bonds that are polarized to be delta positive. So they're polarized because this CH can interact with the CO sigma star. And you can see that here. And so all of these CH bonds are delta positive, and that's exactly where the aromatic lines up. If you don't like this picture, you can look at the molecular orbital picture above. So these interactions are highly stereoselective. The best CH pi donors or bonders are these sorts of carbohydrates, whereas glucose is less good. These data are from just monitoring interactions with straight up indoles. And you can see chemical shift perturbations indicative of binding. And so these beta galactose and beta mannose are the best binders. And we don't see binding with a lot of these other sugars. Doesn't mean they don't occur, but they're not as strong. We could show that this binding is not just hydrophobic, it's stereoelectronic, and we could do that using a Hammett plot. So electron withdrawing groups completely abrogated binding based on our NMR data, whereas donors had enhanced binding, just like you would expect from a electrostatic model. So, and what we saw recently, interestingly, if you look at the data, we saw much more tyrosine in the binding site. But when we initially did calculations, there's not a big difference in tyrosine versus phenylalanine. But in many of the carbohydrate binding sites, that tyrosine residue is anionic. And when you use a deprotonated tyrosine, you can see that there's much stronger interactions based on our calculations and based on the data. The other thing the data shows is that there's no orientational effects. So the tryptophan nitrogen can be in all different directions. And that is really beneficial for recognition of different carbohydrates and actually for sliding of carbohydrates in enzyme active sites, something that we also have seen in looking at a lot of the structures. But this, I think, is also beneficial for those who like to think about catalysis. And there's really beautiful work from the Jacobson lab in which they've used these sorts of CH pi interactions to control structural interactions. And use this catalysis to highlight specific sites. So I'm going to go ahead and just move back to a little bit more of how we've de-orphaned some of these receptors. So if I show you this picture, you can see, again, these sorts of CH-pi interactions enhanced here by this calcium and the Gauche effect. And I told you humans have two of these intellectins, one that I've been talking about a lot, but another one that actually lacks part of this aromatic box. And intellectin 2 really caught our eye because we wanted to understand how changing of a single amino acid might change recognition. And so the two people that took this on are Deepsing Sangtang and Amanda Dugan in my lab. And they were interested in this protein because it's upregulated in pathogen infections. And so when we did use the same sort of microarray, we found that intellectin 2 also recognizes galactose, but now not the furanose form that's only found in microbes, but a pyranose form. And so we were really curious about what it does. So the first thing we did is ask, is this cytotoxic? And so we found that no, it is not. Many lectins themselves are cytotoxic. And by the way, it's okay to eat plant food that have lectins in it, even though there are some things on the internet saying you shouldn't eat things with lectins in it. They will largely be killed when you the lectins will be denatured when you eat them. It's just you shouldn't eat a lot of raw soybeans. And I think no one is going to do that. So what is the benefit of binding to these mammalian sugars? And so we started to think maybe the benefit rested in the mucins. So mucus is composed of glycoproteins, of which I've shown here. They have a folded N terminus, a folded C terminus, but a middle region that's just highly glycosylated and often very extended because they have a lot of prolines along with serine and threonine residues that are extended. And they often terminate in galactose residues. So they have complex carbohydrates attached that have galactose. And so we were curious about whether or not these proteins, intellectin 2, might cross-link mucus. And so we first tested and showed that it bound to the mucus. So this is just a dot blot, and black indicates binding. And these are three mucins, glycoproteins that are found in our gut. And so then we also showed that when you added this protein, it would cross-link these mucins further. So it clustered them, changing their absorbance spectra. And we showed that the same thing occurs when these proteins cross-link polymers. So we knew that the binding is dependent on the carbohydrate, not on the backbone polypeptide. So this shows in blue intellectin 1 and in this wheat color intellectin 2. So very similar structures. Maybe intellectin 2 has a helix, but not so different. So we want to ask, does this lectin bind bacteria? And we showed that intellectin 2 does bind bacteria. So we isolated fecal samples from humans, and then we tested whether or not it would bind. But we also tested specific species. And you can see here that this specific species, the protein interacts with the bacteria, but as we added more and more protein, you saw less of the bacteria bound, and we started to see this kind of waste product. So this is data where each cell is a little dot, I like to say this is like TLC for cells. And so you can see in our TLC that we had the shift to junk, um, usually baseline junk. And that was indicating that maybe the cells were dying. And so we then looked at that and could show that this protein was killing these cells. So it appeared to be cytotoxic. And so we just monitored this. So this is just one bacteria. It binds with no lectin, but then when you add the lectin, shown over here, you can see all the cells just start to pop open. So this protein is really cytotoxic. So we have a protein in us that binds to our mucus and kills bacteria that might cross the mucus barrier. So we asked, does it kill pathogens that infect us as well? And so we saw that it can kill Staph aureus, including methicillin-resistant, antibiotic-resistant Staph aureus. It kills that. So that was interesting because that's an important human pathogen, and it's a gram-positive bacteria, like many of our commensals. So we then tested a gram-negative bacteria. These are much harder to kill with traditional antibiotics, and we could show that this lectin also kills those bacteria. So this, I think, is really exciting because it suggests that we have an endogenous protein that can kill these bacterial pathogens that threaten human health. And that change, I should have mentioned, interleukin-1 shows no such activity. Even though it's very similar structurally, it is not cytotoxic. Interleukin-2 is the cytotoxic one. But the fact that changing a single amino acid in the binding site changes the binding specificity suggests that we could use these as a platform to engineer new lectins that would have specific activities against targeted pathogens or other cells. So I like to say that this lectin is special because it can do both offense and defense. It can protect and cross-link the mucus to function on defense and protect us, but it can also kill the bacteria that might be trying to breach that barrier. And so in this way, it plays these dual roles. So I want to just point out that we need new antibiotics. And one of the strategies that we like to think about is using small molecules to inhibit essential enzymes. But another strategy we think is to think about using these endogenous human proteins to actually control antibiotic, pathogen invasion. So interleukin has these interesting roles in that it regulates our commensals. And I like to think of the need to really tend a microbial garden in our body. And we have these natural proteins that help us do this. And so I'm really interested in further understanding how these proteins function to recognize glycans and how we can co-opt these functions for new diagnostics and therapies. And I'll just leave you with this picture that Jill Wildonger, one of my fantastic postdocs, made to emphasize that all of this depends on really a molecular understanding, and we personally love CH-pi interactions. So I have the privilege to work with an amazing group of students. As you all know, everybody contributes to the project, but I've highlighted really Amanda Dugan, Deep Singh, and I want to highlight Ali Keyes, who did all of the calculations that I showed you, And she was a joint student with Heather Kulik's lab. Katerina Ribik provided the mucins, and then Deb Hung, Romnick Xavier, and Deck Wolfson also contributed to the projects that I showed you. We could not do any of this work without funding. The funding from federal agencies is so, so critical. And please, everyone who's listening, who can make a difference, make sure that all of that funding stays and that it's fairly disseminated. I'll just end there. Happy to take questions if there are any.
Thank you, Laura, for an excellent talk. That was magnificent. We have a first question, and I invite everybody watching and listening to please ask questions. That includes for Christina. We now have the time to do so. The first question is actually from Christina, Laura. And well, did you want to ask your own question, Christina?
Sure, and I was just typing a second one. But oh yeah, but my question was if it is really beautiful, especially with the uh, basic physical organic associations, and it made me wonder, and I was wanted to hear your thoughts about if this helps explain why the hydrophobic effects that you have observed helps explain why tryptophan is the lowest frequency amino acid in the proteome. And
Yeah,
it has this privileged function for binding ligands that are like glycans and others.
Yeah, I think I think that's a good hypothesis because it can have non-specific interactions, right? And that's what you would worry about. It's interesting, it's mostly buried, but in these glycan binding sites, it's on the surface. And as you know, it's also on the surface of many protein-protein interaction sites. And so I think, we use it kind of sparingly to pull out these interactions when we have large surfaces. Yeah. So, and we do see that it is electronically dense as well. And so that's another, I think really critical role that it's not just a hydrophobic residue because you might be able to use right phenylalanine for that, but because it has the electron density, it can interact with these other substrates. And because you brought this up, one of the things that I think about is that we are interested in protein carbohydrate interactions, but actually those who are interested in ligand interactions in general, I think exploiting CH pi interactions and maybe acidifying key protons that are near aromatic residues could be a general strategy. And in fact, that may be what fluorination is often doing and how it's contributing. And so I think that that's an underutilized aspect of molecular recognition.
Can I ask a second question, just also about the tryptophan conservation in lectin, and just have you looked at the conservation in different lectins, does it what your observations are there in terms of the
Yeah,
association with pathogenicity and so on? Yeah.
Right. It is highly conserved. And it is, and its placement can help align where sugars bind and right, which sugars bind where. So, right, I mentioned some sugars, it's much better to stick over a tryptophan than others. And so I think this can help with docking and other things. One thing I also think that's not very been as exploited is putting it in active sites of antibodies could make better carbohydrate binding antibodies, especially if you use it in a very judicious way. So I think about, tryptophan scans. Yeah.
Mhm.
And I didn't have time to put those in, but I think one of the most remarkable things, we've looked at a lot of enzymes that act on carbohydrates, and they just have like these sort of tryptophan faces or aromatic faces that are just beautiful that go along the carbohydrate chain because it allows you to slide.
Mm-hmm. I like that tryptophan scan idea, like glycose scan, but with tryptophan.
Yeah, I think
Yeah.
it could be used much more frequently. Actually, I had a question for you too. Can I ask?
Sure.
Okay. Um, so I just had a question about whether or not you thought there's this glufosinate, it's like a herbicide that
Yeah.
is, right, like an inhibitor of
Mhm.
glutamine synthetase. So I thought maybe do you think that that is its might be part of its role? Like that people just thought, oh, it inhibits an essential enzyme, but it may be actually interfering with this quality control issue.
Oh, yeah. It could like in terms of being able to destabilize the complex
Yeah,
of gluten
I was
synthesis.
thinking maybe its mechanism of maybe you are discovering its mechanism of action where we didn't really know. Because we always just say, oh, you inhibit an essential enzyme, but not all essential enzymes are good targets for drugs. And
Mhm.
so, anyway, I just thought
Yeah,
it was cool.
we haven't tested any glutamine synthetase inhibitors for how they affect the the regulation of glutamine synthetase in terms of its turnover. I always just assumed that glufosinate is mimicking the active site, is
Yeah,
acting as the active site inhibitor, but that would keep the pentamer together. But you're getting at a question that we haven't figured out how to fully resolve, frankly, is when the cyclic imide event is generated and then promotes the degradation outcome, is it the pentamer or is it the multimer that's getting degraded? Is the
yeah.
single unit getting it taken out? And that's been done a little bit, I think, with some of the work that Deshay's lab kind of tried to look at with the N-terminal version, but we haven't looked at that yet in our hands. So
I guess I was
I'll have to take
thinking
a
maybe
deeper look
it's,
at.
yeah, I was thinking maybe it's a 1-2 punch, like, yeah, you have the inhibitor
Mhm.
and you have
Yeah,
degradation.
yeah, yeah. I will definitely think about that more. Thanks, Laura.
Well, Christina and I will just have coffee and talk to each other so we can
Well,
let
this is
everyone go.
absolutely
Yeah.
wonderful. Monica, all of us are sitting by delighted that this kind of, scientific discussion and collegiality could develop very spontaneously in a recorded event, but that's okay. It'll be for posterity as well. This is a great exchange. I myself, when you were talking about the lectins, Laura, was intrigued. We had seen those advertisements, and sort of the science that it's not healthy to be eating foods that contain too many of these proteins. So that was a nice little add-on. Beautiful presentation,
Yeah.
The
both
raw
of you.
soybeans hit home.
Monica, did you have
Please,
something?
everyone eat your
As,
vegetables.
as a
Yes.
vegan, I approve. You should not be eating raw soybeans. So
Yeah.
now this is absolutely excellent. And I was texting Layla and other people that it was a great, discussion going on here between the two of you and obviously the talk. So I don't see any additional questions right now on the panel. Maybe people are pondering or having as much fun as Layla and I are. But just in case to remind people, especially those that had to connect a little bit later, that next week on the 14th, Wednesday at 12 noon Eastern, we are going to have the virtual symposium. Uh, and people should definitely not miss it. So it's going to be via Zoom and also transmitted YouTube. So if there are any additional questions, comments.
Lastly, I wanted to thank everybody, our speakers, Laura, Christina, for joining us and beautiful talks. Also the CIC, the Catalysis Innovation Consortium, and Alan Bailey at the head, of course, with alongside Hugh Davies, our entire committee, virtual symposium committee, Scott, Alex, and of course our Steve, who had to leave, and Amy, our past chair. Looking forward to wonderful Division of Organic Chemistry events this year. And we are absolutely honored to have had you both as our second annual EWOC DOC Symposium speakers. Have a happy new year and take care. Thank you.
Thank you for.
The recording. Thank you. Bye. Thank you, Alan.